Biology, Unit 2, Paper 2, June 2015, past paper time. Here we go. Question 1. Figure 1 provides the rate of photosynthesis in the leaves of a plant species measured in response to light intensity. Let me look at this here. On the x-axis, we have light level, which is the percentage of full sunlight. And we have the photosynthetic rate in cm cube, oxygen per hour. At zero light intensity, we have no photosynthesis occurring, right? So when we put a little bit of light, we get a little bit of photosynthesis occurring. And it's occurring at a pretty much a linear rate where you see in A. And as light level increases, so does the photosynthetic rate. So at this point A, this is where we'll say light is a limiting factor, because the amount of light is actually limiting the rate of photosynthesis at this point. If you increase the light intensity, the rate of photosynthesis increases. So this stops being linear after about 20%, and you'll see at around 70% or so, between 60 and 80, that the photosynthetic rate sort of plateaus. It becomes constant. This is because at B now, this is because light is no longer a limiting factor. What does that mean? That means that all the electrons are already stimulated in the photosystems, and that means that if you increase the intensity of light, if you increase the percentage of full sunlight exposure on this leaf or this plant, the photosynthetic rate is no longer increasing, which is what you're seeing here as a plateau. It's constant. That means that some other factors become limiting. Could be carbon dioxide or water. So the question here is, describe the observed effects of light intensity on the rate of photosynthesis of the species in the regions labeled A and B. Where's what we just talked about. So A, light is a limiting factor. Increase in photosynthesis rate is directly proportional to increase in light intensity. That's why it has a linear relationship there. And B, light is no longer a limiting factor. The rate of photosynthesis becomes constant as light intensity increases. Just 2 marks. So just a basic explanation would be okay to get 1 mark for each one here. Part 2. Outline the process by which oxygen is produced during photosynthesis. This is a 2-mark question, and let me explain it here. So to explain this here, we'll see that we have 2 photosystems. Photosystem 1 PS2 and photosystem 1 PS1. And what we're going to have is an electron, and this is what you call if you follow the orange arrows, this is non-cyclic photophosphorylation. That basically means addition of phosphates using light, right? Photo means light, phosphorylation means addition of phosphates to a molecule. And we will see here that light stimulates photosystem 2 to release an electron. That electron sort of goes like a pinball in a pinball machine and goes clink, 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 clink all over the place, but really is bouncing off of different complexes. It goes to photosystem 1. You see ATP is produced and bounces off here again. 2 electrons convert a coenzyme NADP into NADPH. We don't need to talk about that. But basically what this electron is used to do is to provide energy for photolysis. Photolysis is the splitting of water molecules into hydrogen and oxygen. So that's really what we want to talk about here. So light energy causes electrons to be emitted from photosystem 2. These electrons return to photosystem 2 from water, and this releases oxygen, from water molecules, photolysis. So let's look at B1 using an annotated diagram of a simple respirometer constructed from a plastic syringe and capillary tube. Outline the procedure for measuring respiration and germinating seeds. This is our setup of our apparatus here, and this is the plastic syringe that you're seeing here. Inside the plastic syringe, we have some germinating seeds. These germinating seeds are of course going to be exchanging oxygen for carbon dioxide, but we have soda lime for absorbing the carbon dioxide, so it doesn't really affect the experiment too much. But as oxygen is taken up, it's going to start pulling on this little capillary tube here. What's the significance of that? But what we have here is a little bead of colored water, right? And this might be red or blue, is basically so you could have higher visibility. And as oxygen is being taken up, you're gonna see that this little bead here is gonna start moving along the capillary tube. Now, what you're gonna have here is not really so much depicted here, but what you're gonna have here is a little piece of graph paper or ruler to measure the distance at which this colored water bead is going to move. So you'll also have a stopwatch in here to measure time intervals. With that, there you can actually measure the rate or the speed by measuring the average distance over time at which this colored water moves. Now if we look at the procedure here, this is what we do is, so our diagram might be about 3 marks, and we just need to put a simple procedure here. What you're going to do is you're going to place the weighed seeds in the respirometer, and remember these seeds have to be alive because they have to germinate, and which means that they have to undergo gaseous exchange. You're going to introduce a colored water droplet into the capillary tube by dipping the tip of the by dipping the tip of the syringe in a beaker filled with water. So it just has to be a small water droplet. Next, you're going to record the movement of the meniscus of colored water drop over time to calculate the volume of oxygen intake. Or do you record that? Use the ruler or the graph paper as seen there. So the next part here is to suggest 5 precautions to be taken to ensure the accuracy and reliability of the results when conducting an experiment using a simple respirometer. Well, I have 7 here, so any 5 of these would work, and it's just 5 marks, so it's basically 1 simple precaution per mark. 1, you're going to ensure that the apparatus is airtight. Okay? You don't need any atmospheric pressure affecting the experiment. 2, you're going to ensure that the germinating seeds do not touch the soda lime. That could basically affect the experiment. 3, ensure that no air bubbles are in the capillary tube with the fluid that will affect the movement of the bead itself while you're trying to measure distances over time. 4, use fresh, healthy seeds and try to make sure that they're from the same batch. 5, repeat the experiment and determine the averages, basically a staple of any precautionary experiment. 6, place the respirometer in a room with constant temperature. Basically, temperature affects rate of uptake of oxygen. It affects the respiratory process. So you don't want any fluctuations in temperature. You don't want to have it in a very cool air-conditioned room where respiratory rates would slow down. 7, you're going to set up a control respirometer with a non-respiring substance. You could use like beads or something, for example. This is just to ensure that the syringe is working fine, that there's nothing in the room externally that is affecting the experiment or the results of the experiment. Any 5 of these would give you 5 marks. Let's look at question 2. Figure 2 is a diagram of a simple potometer which is used to measure transpirational rates, which is used to investigate the rate of transpiration of leafy shoots. And here we have a potometer. We have the capillary tube is being dipped into a reservoir through capillary action. This is going to start taking up water. We have a ruler to measure the rate over time, and we have a little air bubble here that will move when water is taken up. That is used as basically the marker to measure the rate. Now, on figure 2, use an arrow labeled X to indicate the position at which a leafy shoot is placed on the potometer. X will be right here where you see this, it has this sealed bung, and the plant will be placed here where the roots will be dipping into the water, where the water could enter the plant via osmosis and then into the xylem via transpirational pull. 2. Outline the key steps for determining the rate of transpiration using the potometer shown in figure 2. Well, 1, you're going to measure the distance traveled by the bubble in a given time interval. Could be 30 seconds, 20 seconds, whatever you pick. 2, take several readings several times for the duration of the experiment. So you can measure the position of the bubble at 10 seconds, at 20 seconds, at 30 seconds, at 40 seconds, and so on. And measure the distance at which that bubble, or the displacement, I should say, from the initial position where that bubble has reached. And 3, you're going to calculate the average reading, and the rate of transpiration would be obtained from the average distance traveled per unit time. Think of that as the speed of the bubble, and thus the transpirational rate. Let's look at B. With reference to 4 structural features, explain how xylem tissue is designed to facilitate water transport. You don't need to define transpiration or explain the actual mechanism of water transport, so you don't need to explain transpirational pull and root pressure and things like that. You're just sticking to the facts about the xylem. So 1, the xylem tissue has a narrow lumen. The lumen is the space, imagine like the little gap inside, or little space inside where the water will actually pass through the tunnel. That facilitates transpirational pull due to the adhesion of water molecules. Now, because it's narrow and water molecules tend to stick to other substances easy, that's called adhesion, the narrow lumen actually helps that out. 2, the xylem is not alive. It actually contains rings of dead lignin, and as required for support. Lignin is quite rigid, so it helps the xylem stay upright while all this water passes through it. 3, the xylem vessels are hollow, which means that there is no cytoplasm in them, as they are not alive, and that allows rapid transport of water. And 4, xylem vessels are elongated and arranged end to end to form a continuous column. So they're not like phloem tubes, which have perforated ends or sieve plates. Xylem vessels are continuous, which means that again there are no end walls. So water can have basically a rapid constant flow through it. Each one is 1 mark. Alright, let's look at C. Figure 3 is a student's plan drawing of a cross-section of a major mammalian artery showing the distribution of the tissue layers. And the question here is to you say in the region highlighted by the box label A, this is the box A. On figure 3, draw cellular details of the tissue layers. So what you're gonna do is you're just gonna draw, you don't have to redraw it, but you draw it inside this box here. And there's 4 marks, so you're gonna have about 4 different points here. And inside here we have the lumen. So there's the lumen of the artery, there's where the blood does flow. The endothelium is going to be nucleated. You're going to have little nuclei here, so you're going to make sure and put a few dots in here. And right above the endothelium with the nuclei, you're going to have the muscular layer. So this is the media, which is the smooth muscle. These basically contract and relax as blood flow occurs through the artery. And that actually allows high blood pressure because they could withstand the pressure. We also have the elastic layer between the endothelium and the media, and this facilitates the stretching of the arteries as well, so that they don't burst. And lastly, on the very outer layer here, we have the media externa, or you could call the adventitia. This is another muscular layer, but also has collagen or connective tissues in here. Part 2. With reference to the structure of features observed in figure 3, explain how an artery is able to endure the high pressure of blood as it is pumped from the heart. Well, just explained it there. Arteries have very thick walls which stretch under pressure. They are made up of smooth muscle and elastic fibers, which again can withstand high blood pressures. 3. State one major difference that would be observed if a student did a plan drawing of a cross-section of a vein. I'll pop a little diagram, a little micrograph here, and you'll see what the artery looks like compared to the vein. You'll see that the vein actually has a more irregular shape. It's not as round. It actually has a more oval, elongated kind of look. The lumen is larger and has a more elongated, irregular shape, and they have much thinner layers of media or muscular walls. Let's look at question 3. A. Table 1 shows the percentage of young people in a population who were categorized as being obese over the period 1971 to 2006. Here we have the table. Now on the table itself, we have time periods 1971 to 74, 76 to 80, 88 to 94, 2003 to 2006. We have 3 different age groups depicted: 2 to 5, 6 to 11, and 12 to 19. What you're seeing here is the obesity levels and percentage of young people. So you're seeing the 2 to 5 as the years, basically for all of them, by the way, as the years progressed, the rate began increasing, at least at different rates, because you see at 2 to 5 it went from 5 to 12.5 in a span of, maybe say, almost 30 years, right? 6 to 11 it went from 4 to 17, and 12 to 19 it went from 6 to 17.5. So that means that the 2 to 5 age group had the least increase in obesity levels over the period of time, but all of them increased. This is what your bar graph will look like. You want to have them in triplets like this per time period. Make sure that you have a key or legend. So you could use different patterns. You could use dots for one. You could have one plain, and you could have one hatched or striped. And make sure to have gaps between the datas. 2. Describe the changes in obesity levels among the age groups over time. We just mentioned this. There's a general increase in obesity over time for all age groups, but the increase in obesity for the 2 to 5 age group is less than the older age groups. 1 mark for each one. Suggest 3 possible reasons which may account for the trends described in A2. So why is the obesity levels of children increasing? And well, one could be lower levels of physical activity over time, as possibly due to the advent of electronic devices. It's made available widely now, and that kind of take over the lifestyle of playing outside, things like that. 2, you have poorer eating habits, because at least in the 70s and 80s, you had the rise of fast food and junk food, and being made more accessible for families, especially those of lower income brackets. So there's also socio-economic reasons where more poverty could lead to buying more fast food. So there's a lot more franchises that have sprung up over time, and that could have contributed to obesity levels. Let's look at B. Figure 4 shows the concentration of antibodies in the blood of an individual during the first few weeks after an infection. And on this graph here, we could see the time in weeks. 3 weeks has passed, and we have an infection at week 0, and this shows the y-axis shows the concentration of antibodies in the blood. We could see after the infection that there was a small delay before the concentration of antibodies began to sharply rise. It reached a peak right before 2 weeks, before the concentration of antibodies would have decreased. Remember, antibodies would have helped to eradicate whatever pathogen or antigen would have caused this infection in the first place. So they say describe the response shown in figure 4. And it's 2 marks, so description of just the shape of what's going on. So there's a rapid increase in antibody concentration a short time after infection. You could also mention that there's a small lag time before the response and the detection. So in about 2 weeks, it reaches its peak level before slowly decreasing. Part 2. Explain whether this response is likely to be a primary or secondary response to the infection. Now what we have to look at is the steepness of the curve with relation to the concentration of antibodies, because it's quite steep. The amount of antibodies has been produced very, very quickly, and this is most likely due to a previous exposure. Due to previous exposure, you would have a lot of cloned plasma cells that would have already been there in the bloodstream. You would have memory cells that would have retained information about the pathogen before. So this would be a secondary response due to the short delays, about 2 days, and memory cells may have been present from an earlier exposure. It's just 1 mark. Alright, so let's look at C. Distinguish between active and passive immunity. So active immunity comes from antibody production due to a previous exposure to a pathogen or antigen. Basically, you get infected and you produce antibodies and memory cells, so you're able to fight off the infection faster in the future. Now this could be unintentional, which means that you're just walking outside and somebody sneezes out some viruses and you breathe it in and get sick. Basically what's happened with COVID. Or it could be intentional. You can inject a weakened version of the pathogen into your bloodstream and enact an immune response from that, and that would be vaccination. Passive immunity, however, is when it is provided externally to the body's immune system. The body doesn't really expend energy to produce antibodies to get it from outside. So it occurs from antibodies provided externally or artificially. So it could be from your mother via the placenta or via breast milk, or it could be injected, or it could be through monoclonal antibodies through some kind of serum. Let's look at 4. 4a. Feeding relationships and the transfer of energy through the biotic component of ecosystems have traditionally been summarized in pyramid diagrams. 1. Explain why energy transfer between trophic levels is considered to be relatively inefficient. In your answer, provide a brief definition of the term trophic level and include 3 reasons for this inefficiency. So let's look at this. A trophic level is a group of organisms that occupy the same feeding position in a food chain. So in a food chain where the producers would absorb the solar energy from the sun, and then you'd have your primary consumer, your secondary consumer, your tertiary consumer. Each of those there is called a trophic level. Only approximately 10% of energy is transferred from one trophic level to the successive level. So this is true because let me say, for example, that a rabbit is eaten by a hawk. The hawk might only get 10% of the energy from the rabbit. Why is this? That's because the rabbit had to spend or expend 90% of the energy actually undergoing its metabolic processes. The rabbit had to grow, the rabbit had to respire, the rabbit had to move around, the rabbit had to reproduce, the rabbit had to respond to different environmental pressures. And that took about 90% of the energy. So when the hawk ate the rabbit, it will only get about 10%. So where does that 90% go? That 90% is actually lost to the atmosphere as heat. So reasons for this inefficiency, well, I'll just explain it. Energy is lost as heat during respiration and excretion. Energy is used by the organisms for metabolic processes, such as growth. And some of the organisms die and are not eaten. So if you use the same example with the rabbit and the hawk, well, you have all these rabbits running around, and some of the rabbits might live to old age and would never be eaten. So that energy would stay with the rabbit and would never move forward in the food chain, if you can understand what I mean. Same thing with plants. Think of most plants. Most plants in the world are not eaten. So a lot of that energy just remains in the trees or in the plants or in the leaves. That's a good thing, by the way. Part 2. Briefly describe the 3 types of biological pyramids and comment on which type is the most useful for representing the productivity of each trophic level. This is 5 marks. So we have 3 types of pyramid: the pyramid of energy, pyramid of biomass, and pyramid of numbers. The pyramid of energy represents the amount of energy at each trophic level. Biomass represents the dry weight of organisms at each trophic level, and numbers represents the number of organisms at each trophic level. This should be of numbers. Now, pyramid of numbers, again, is just tells you at that particular time it have 5 hawks in this area, it have 200 rabbits in this area, it have 1,000 shrubs in this area. So it just tells you the number, but it doesn't actually relate to the weight or the dry weight or the biomass of the organism. That's where the pyramid of biomass will come in, and this would relate more to the actual amount of organic material that is found within the organism. The pyramid of energy, however, represents the amount of energy that could be passed through organisms or members of each trophic level. So the pyramid of energy is the most accurate representation of productivity, and this is actually because it's not limited to one point in time. Numbers and biomass are limited to one point in time, but energy more takes a look over time, if you understand what I mean, because it takes at large populations on the whole. It doesn't really depend on the number of organisms. So it also shows the actual energy transferred and includes the solar energy input. B. There's a continuing debate as to the nature of the relationship between biological diversity and ecosystem stability. Outline the current view of the nature of this relationship and briefly discuss evidence in support of the stated viewpoint. This is 4 marks. So biodiversity basically says that they are great. It relates to species richness, if you can think of it like that. If there's a high number of species in the area and thus a high number of species interactions, that they're saying that contributes to ecosystem stability. So high biodiversity promotes stability of ecosystems with complex food webs. Basically, more and more feeding relationships, more and more symbiotic relationships. Increased complexity is usually associated with greater stability. So an example would be in the forests of Indonesia. They usually cut down a lot of these natural forests in order to plant palm trees to produce palm oil. Palm oil is actually used in a great number of commercial products. I'm gonna name any here, and you'll find them in almost everything. So it has a very high demand. And because now a lot of this deforestation has happened, there's a lot of destruction of organisms' natural habitat. So it has greatly reduced its species richness, the forest itself, with several fauna being in heavily reduced numbers such as elephants and orangutans. A lot of birds have actually migrated because the homes have been destroyed. So this would be one example of how low biodiversity actually decreases stability, because organisms would actually migrate as a result. If trees and flora and so on get removed, or if certain animals get removed, that would affect the entire feeding relationship, the entire food web. So another example would be the Serengeti National Park. This has a very high species richness, and over time this national park has been studied and the ecosystem has remained stabilized for a long period of time, at least without human intervention. Let's look at question 5. A. Explain the mechanism for selective reabsorption of glucose from the glomerular filtrate in the kidney, including the role of sodium ions. Discuss the clinical significance of the presence of glucose in the urine. Okay, so I'll pop a little diagram just now to explain, but let me read here. Reabsorption of glucose occurs in the proximal convoluted tubule of the nephron. Now, this occurs through sodium-potassium pumps, which are usually used to maintain a resting potential in axons, well, neurons. So whenever sodium ions are transported out of the tubule cells, glucose is co-transported into the bloodstream. Let me show you what that means. So to explain this here, this is a cross section of the nephron, well, of a part of the proximal convoluted tubule. Here we have the epithelial cells, and here we have a capillary. We have some interstitial fluid here. Along the epithelial cells, we're going to have some sodium-potassium pumps, and those are usually used to maintain a resting potential, which we're not going to get into here. But what typically happens here is that sodium is going to be pumped out of the proximal tubule cell, and potassium is going to be brought in. And this occurs through the sodium-potassium pumps, and this is actually active transport. Now, glucose, the funny thing about this is glucose is usually transported via facilitated diffusion through specific carrier proteins. So, but the thing is here, when these little pumps open up, when these little proteins open up to let sodium out, it's kind of like glucose find a little, they see a little door open, and it's like, hey, look, that door open, let me go through there instead. So glucose is actually actively transported out, but only because the gate was open when sodium was coming out. So it's kind of strange to think about, but think of it like glucose see a gate open, must be like, hey, the gate open, sodium open the gate, let we go too. In this case here, we see that sodium is co-transported out of the tubule cell. So sodium ions are actively transported out of the PCT cells, and glucose is also co-transported into the bloodstream. And the clinical significance of glucose in the urine. But the presence of glucose in urine may indicate diabetes due to the glucose concentration in the blood being very high. If you already have a high concentration or high solid concentration of glucose in the bloodstream, it means that the rate of diffusion of glucose from the nephron back into the bloodstream would actually decrease. And this could be, again, is a major indicator of diabetes, whether it's type 1 or type 2. But it could also be a problem with the kidney function could be maybe impaired, or it could have, the nephrons could be damaged in some kind of way. Starting with atrial systole, describe the mammalian cardiac cycle. Your answer must include the cycle initiation and mechanisms that prevent reverse blood flow. Well, the cycle is initiated via electrical impulses stimulated by the sinoatrial node, or the SAN. So what that does is the sinoatrial node emits electrical impulses across cardiac muscle, and this stimulates the contraction of atria. The atria are the upper chambers of the heart. So when those contract, you could think of it like they will send blood to the ventricles. So the atria contract, forcing blood into the ventricles. So this is called systole, atrial systole. Ventricular systole. So a way I think about systole, the word systole is like S, I think like it's for squeeze. Imagine like it's squeezing those muscles and the blood flowing down some valves, if you understand what I mean. So ventricular systole causes both ventricles to contract to force blood out of the heart into the pulmonary arteries and the aorta, basically to send blood out of the heart, and so it could go to the rest of the body. Again, it's like imagine you're squeezing the bottom of the heart, and you're squeezing all that blood out into the aorta, so it could get out into the bloodstream. That is ventricular systole. Atrial and ventricular diastole then occur, which is basically the heart chambers relaxing and allowing blood to flow into the heart from the vena cava and the pulmonary veins. So blood flows back into the heart, and the heart gets refilled. So how blood flow, how reversal of blood flow is prevented is that, or backflow, is that you have valves in the veins. And you also have valves in the heart. You have your semilunar valves, and you have your tricuspid and bicuspid valves. Those allow blood to flow one way, but not the other way. C. Explain the regulatory effects of blood carbon dioxide levels and the hormone adrenaline on heart action. So carbon dioxide, high carbon dioxide concentration leads to the stimulation of sympathetic nerves in the body. And what this will do is increase heart rate. And in response, so if you think about it, you have high CO2, you want to get more oxygen to dissipate that CO2, because the CO2 is waste material, right? You want to get more oxygen in. So these CO2 levels are detected by chemoreceptors in blood vessels. Adrenaline, on the other hand, you know it as if you get scared or if you're in a very nervous situation or stressful scenario. During these periods, impulses from the brain stimulate adrenaline production by the adrenal glands. Those are located right above the kidneys. Adrenaline circulates in the blood, targets the heart, it stimulates the sinoatrial node, and basically tells the heart to beat faster and to increase the force of those contractions so that more oxygenated blood could reach the muscles, and so you're able to escape whatever scary thing, or you're able to handle whatever stressful situation that you're in. Okay, let's look at 6. The World Health Organization defines health as being a state of complete physical, mental, and social well-being, and not merely an absence of disease or infirmity. Define the term disease and discuss 3 different types of factors that may influence a person's health. So a disease could be simply defined as a condition that impairs the normal function of the body. It may refer to an illness of the body or the mind. Now, factors that influence health. We need 3 of them. One of them is lifestyle and behavior. Examples of this would be if you have a poor diet, low physical activity, you abuse drugs, or you have general behavior that would lead to exposure of pathogens. In the case of if you're in an area with a lot of COVID positive patients, if you work in a hospital and you're wearing your protective equipment or your mask or anything like that, of course you're gonna get sick. So all of this can increase risk of disease or infection. 2 would be genetics. So diseases such as hemophilia and sickle cell anemia can be passed down from parent to offspring due to inheritance of faulty or mutated alleles. So this wouldn't be restricted to just those that are hereditary. Well, hereditary in terms of inheriting alleles, but it could also be the risk of the disease could be passed on from generation to generation. A very good example is diabetes and certain types of cancers. Lastly, we have the environment of the individual. So a person's physical environment can influence whether or not they have access to potable, that means drinkable water, sewage treatment, and the likelihood of vectors depositing pathogens. If you live in an area that is prone to this next to a swamp, for example, and it has a lot of breeding grounds for mosquitoes, you have a higher chance of getting malaria or dengue, for example. If you live in an area that has a lot of rats, you have a higher chance of getting leptospirosis. If you live in an area where the water is not very drinkable, you have a high chance of getting cholera if you don't treat it. Part 2. State 2 main categories of human disease, giving a brief description of each category. With the aid of an example, comment on the problem of classifying a disease into one particular category. So I have 3 different categories here. One would be deficiency diseases. So those are usually caused by an absence of nutrients in the diet. Example would be scurvy if you have a lack of vitamin C or ascorbic acid, or rickets if you have a lack of calcium or vitamin D, which leads to the bones becoming very brittle. Other examples would be like night blindness and iron deficiency anemia. 2. Infectious diseases, also called communicable diseases, are those that are caused by bodily contact with pathogens, and usually those that could be spread. This also involves every sexually transmitted infection, such as gonorrhea, chlamydia, or syphilis. Mental diseases are disorders associated with brain function. Common example here is schizophrenia. You have other categories too. You have social diseases, chronic, and genetic. Now, the problem with classifying diseases into a single category is that many diseases may overlap into multiple categories. So, example is type 2 diabetes. You can inherit the risk for it, so it could be considered somewhat genetic. But it could also be classed as physiological, which means that it affects an organ, the malfunction of an organ. Chronic, which means that it is a long-term disease. And a social disease, which means that it is usually associated with a kind of lifestyle, a sedentary lifestyle or a poor diet. So that is the problem with classifying diseases in one category. The last question here. Sandra and Wayne are both drug abusers. Sandra, who is 40 years old, uses about $100 worth of heroin per day in 2 to 4 injections, 4 to 12 hours apart. When she goes for longer than 12 hours without the drug, she feels sick in the stomach, becomes restless and anxious, and has very strong cravings for the drug. We can safely say that Sandra is undergoing withdrawal symptoms when she doesn't have the drug, and she has to take it fairly often. In comparison, Wayne, who is 16 years old, sniffs petrol with his friends. He often goes without sniffing for a few days with no ill effects, so he could go a longer time without Sandra, and he doesn't suffer from withdrawal symptoms like she does. However, when he has not sniffed for a week or more, he can feel a strong urge to sniff, despite knowing the dangers of petrol sniffing. The urge to sniff is very strong when he feels depressed or bored. Identify the type of addiction demonstrated by Sandra and Wayne and justify your answers. And it should also include a brief explanation of the term drug abuse. So just to define what drug abuse is defined as excessive use of drugs for non-therapeutic use. And this usually would lead to some form of a highly increased tolerance and addiction. So Sandra, we see, couldn't even go through the day without using heroin. So she exhibits a physical dependence on the drug, and she has very, very strong cravings. And she actually shows withdrawal symptoms such as nausea, restlessness, when use of the drug is abruptly reduced for short periods of time. Wayne, on the other hand, doesn't have withdrawal symptoms. If he has it, is much less. He mostly uses drugs when he feels bored, so his use of it exhibits psychological dependence. He has less cravings, and he has the ability to go longer periods at times without using the drug. A lot of psychological dependence usually comes in social situations, or when it comes from, they could say when it's with peer pressure, or it usually arises when something goes wrong in someone's life and then they have to drink or they have to smoke. That's usually due to a psychological dependence rather than a physical dependence where your body becomes physically ill if you don't actually receive the drug. Basically, you get strong withdrawal symptoms. And that is 2015. Take care.